Hi everyone, we hope you are having a good day. We are going to talk about from the topic of the production and the evaluation of the Fucoxanthin from the Marine Diatom Odontella aurita. From the class for topic of biotechnology, aquatic biotechnology. The teacher is Lourdes Díaz Tenorio and the team is Team Aguachiles.
Fucoxanthin is a major carotenoid in seaweeds and diatoms. This pigment forms, together with chlorophyll A, chlorophyll C, and anoprotein, a major like harvesting fucoxanthin chlorophyll A and C complex, which transfers light energy to chlorophyll A of the photosynthetic reaction centers for photosynthesis. The advantage: this carotenoid exhibits strong antioxidants, anti-inflammatory, anti-obesity, anti-diabetic, anti-cancer, and anti-hypertensive activities. Fucoxanthin can also be used as an animal feed additive in poultry and aquaculture industries.
The article that we are going to analyze is the production, characterization, and antioxidant activity of fucoxanthin from the Marine Diatom Odontella aurita. These are the authors, and this one was investigated in 2013.
The objective from the work is the effects of the nitrogen concentration and light intensity on fucoxanthin production and the commercial potential of the fucoxanthin production in a pilot scale photobioreactor.
First of all, we are going to see some characteristics of this microalgae, Odontella aurita. It's a bobbin-like unicell. It can measure around 15 to 30 micrometers. It has high concentration of fat acids. It can accumulate high concentration of fucoxanthin, which is something very attractive for this investigation. That is the product that they wanted to extract, and they are often found in coastal waters. That makes it a very easy way to obtain the fucoxanthin because they are easy to find and also to extract from it. It can be a very feasible way to do it. The microalgae was obtained from a Scandinavian culture collection of algae and protozoa. This is located in the University of Copenhagen and was maintained in a modified L1 medium. The culture conditions of Odontella aurita were in a photobioreactor with bubbles of about 3 centimeters of diameter, with temperatures around 25 degrees Celsius, and with a control concentration of CO2, or carbon dioxide, of around 1%. Also, the nitrogen media was regulated, one with a lot of nitrogen quantities, and the other one had a deficient nitrogen concentration. The process of extraction of this pigment was very simple. They used 10 milligrams of freeze-dried algae powder. This one was extracted with 5 milliliters of ethanol in a conical centrifugation tube with a stirrer for 2 hours in the darkness. Later, the mixture was centrifuged for 5 minutes at 3,500 revolutions per minute and later was analyzed in a HPLC. In the image, we can see the separation of the phases, each one containing a specific pigment concentration. For the purification, as we mentioned before, they extracted the pigments from a freeze-dried algae powder. Later on, this one centrifuge, and the centrifuge was analyzed with HPLC in a 6-4 concentration volume of N-hexane and acetone, with the silica gel column being the solid phase. The one that we are interested in is a red-orange, where you can see that there is the number 2 and the number 2 is a Fucoxanthin. This one was later on passed to a prep HPLC to purify it even more, to around 96% of purification in total.
Results: to evaluate the potential of fucoxanthin production in aurita, growth experiments were conducted to the 3 cm of bubble column photobioreactor with a nitrogen replete and nitrogen limited culture media. The cultures were subjected to a low and high light intensities. At the low light, aurita exhibited nearly identical growth with two different concentrations of nitrogen, and the maximum biomass concentration of the cultures occurred on day 10, how we can see in figure 1a. At the highlight, more rapid growth occurred in the cultures containing higher nitrogen concentration, and the maximum biomass concentration of 6.36 was obtained on day 10, which was about 50% higher than that in the low nitrogen cultures. Fucoxanthin has a lot of uses. One of them is antioxidant. Fucoxanthin is a good antioxidant and free radical scavenger. This carotenoid can be activated in both hydrophilic and lipophilic environment. Apart from capturing free radicals and preventing lipid peroxidation, fucoxanthin activates its own antioxidant system through the protection of the transcription factor NRF2. The antioxidant activity of fucoxanthin is 10 to 1,000 times more potent than vitamin E, vitamin C, and carotenoids such as lutein, lycopene, beta-carotene, and zeaxanthin. Human studies have confirmed the role of fucoxanthin as a potent antioxidant and anti-inflammatory. Health. Fucoxanthin improves the elasticity of the blood vessel wall and increases the vasodilation induced by nitric oxide, thus lowering blood pressure. Blood viscosity is also improved by fucoxanthin and causes a reduction in triglyceride levels and an increase in HDL cholesterol and adiponectin. Preclinical research indicates that fucoxanthin exerts a protective effect against atherosclerosis. This substance inhibits the inflammatory process of the blood vessel wall and reduces the risk of thrombosis as a consequence of the rupture of the atherosclerotic plaque. Several investigations with experimental animals have shown that fucoxanthin improves insulin sensitivity and glucose metabolism in a diet high in fat and fructose. This improvement in insulin sensitivity is caused by reduced oxidative stress and inflammation. The use of fucoxanthin supplements lowers the risk of insulin resistance, leading to type 2 diabetes. Fucoxanthin protects the beta cells of the pancreas from damage caused by oxidative stress and inflammation caused by the resulting high glucose level of insulin resistance. In addition, fucoxanthin protects the kidneys from the type of glucose-induced damage. Hepatic protection. In experimental animals that consumed obesogenic and inflammatory diet, the supply of fucoxanthin reduces hepatic steatosis. This type of diet consists of a high amount of fat and fructose. Fucoxanthin counteracts hepatic steatosis through, among others, potent inhibition of endoplasmic reticulum estrase.
For the conclusion of the work, the production of fucoxanthin from the microorganism is very attractive, with the maximum yield of 79.66 mg for liter, less one, achieved in the bubble column in the photobioreactor. A comparable yield obtained in the scale of flat plate from the photobioreactor confirmed the technical feasibility and the scalability of the microorganisms. The purified fucoxanthin was identified as a trans fucoxanthin and showed strong antioxidant properties. These results suggested that the microorganisms may be a promising natural source of fucoxanthin for the human health.